Our next speaker is Dr. Billy Li. Dr. Li is a professor in the Department of Genetics at Stanford University and a member of BioX. He received his PhD from Washington University in St. Louis here, working in the labs of Susan Dutcher and Gary Stormo. And after completing a postdoc fellowship with George Church at Harvard University, he joined the faculty at Stanford University. His lab focuses on RNA editing mediated by ADAR enzymes and its application in gene editing technologies as treatments for cancer and autoimmune disease. In 2020, Dr. Li received the Chen Award of Excellence from the Human Genome Organization. Please welcome Dr. Li.
Well, what an honor to be back. Thanks, Ting and others for putting this amazing symposium together to honor Gary. Yeah, so maybe before I start to talk about some of the research that is currently ongoing in my lab, I just want to give you a kind of brief overview of my academic journey. Before coming to the US in year 2000, I graduated from Tsinghua University in Beijing. As many of the other fellow immigrant scientists, I would say it wasn't easy for sure in the beginning. I remember taking maybe the nucleic acid class in this room. I told Kathleen Hall, maybe last year when I was here, I barely understood anything she was talking about. That was who I was. And then luckily, I also got to WashU via this Monsanto fellowship. That is Monsanto, is a local company headquartered in St. Louis. And I was also very lucky enough to be co-advised by Susan and Gary. I know there are a lot of scientific children of Gary, but I guess I may be most kind of qualified to say I'm the scientific child of both Susan and Gary. That may be why I'm standing here to speak to you, representing a lot of amazing trainees of Gary. And then after I graduated, soon I moved to Harvard Medical School and did my postdoc work with George Church. And that was January 2006. And then in September 2010, I moved to Stanford to start my own lab that focuses on RNA editing. I will talk about later. All right. So during my 5 years at WashU with Susan and Gary, I was just so lucky to be able to have this exposure of combining computational biology and experimental biology, or wet or dry type of approaches. So I guess also I was just so lucky to enter at the right time. And when Susan's lab was able to generate a small fraction of the genome of this single cell organism called Chlamydomonas, I remember that was less than 1% of the whole genome. And I was tasked to just annotate that part of the genome. Although it was a small percent of the genome, I was able to learn so many computational tools, such as gene prediction, such as estimating the total number of genes that may be available in this genome that wasn't even available. I was so lucky to got some help from Takis and others, and Takis is going to speak next. So it's really great to be able to learn a lot of things computationally in the beginning. That prepared me very well to tackle probably a more important question that is the main thesis of my work that was eventually published in year 2004. So in this work, what we did, this is kind of one page of the paper, maybe for students, you cannot imagine you can have figure 1 with one Venn diagram in a cell paper. That was what happened. I just want to zoom in to you. That's figure 1. That's the entire figure 1. So in this paper, well, this is actually the key figure, I would say, of the paper. So in this work, we were lucky to use this very simple comparative genomic approach, obviously it's a computational approach, to look for genes that are conserved between the single cell green algae, clammy, and humans, which is a very complex organism. But these two organisms obviously are very, very different. But one thing they do share is that they all have cilia or flagella in Calaminomonas. In humans, that's called cilia. But we also know that land plants, such as Arabidopsis, do not have flagella or cilia. So by doing a simple approach, but maybe different from most of the comparative genomics approach, to look for the things that are shared across everything, we're looking for things shared between Clammy and humans, these two ciliary organisms, but not present in land plants that do not have cilia. So by doing that, we were lucky to have only more than 600 genes. So then it turns out this set of genes appears to be very sensitive and specific in terms of finding these ciliary and basal body genes. And basal body essentially is the base of the cilia. So that was really a unique case, I would say very lucky case, to use this very simple computational approach to identify a lot of important genes. And in this paper, we also found one of the human disease genes called Bardet-Biedl syndrome 5 gene. So I want to use this example to say, it really made me think computation can be the driver of a project. Because typically we think about computational analysis is just to analyze data, so that you get something out of that, which is very helpful. But in this case, we actually use computation as the initial driver that, as a foundation to do more interesting things. And obviously, we also learned computation is not everything. We can also combine that with many other approaches. Then, of course, we got some press from this Outlook journal published by WashU back in 2004. This is the time we all look younger. Also there's a technician Susan that Linya also contribute to the work. And then one of the things, when I was a student, I went to Gary's lab for weekly lab meetings. So one of the things I didn't expect to learn at all was to learn from a fellow graduate student, actually the same year, Ritesh Agarwal. I think he was working on something, looking for RNA editing sites using worm EST sequences. For younger generation, you probably haven't heard of EST. Essentially, that's RNA-seq data with longer reads, but small number of reads called express sequence tags. And then Ritesh, I think, was working maybe either the data from Brenda Best's lab or collaborated with Brenda. I remember he mentioned Brenda Best in his talk in the lab meeting. Essentially, he was just looking for RNA editing sites in worms. So what is RNA editing? So I realized there's something called adenosine to inosine, or A2I, that is a deamination reaction catalyzed by this enzyme called ADAR, adenosine deaminase, acting on RNA. So inosine pretty much looks like guanosine. So essentially, it's A2I, sometimes people call A2G changes in the RNA. So that's kind of the exposure. Obviously, I didn't know much about it. I just heard about it. And then I moved on to George Church's lab, not expecting to study RNA editing at all. So the reason I moved to Boston is really because of the revolution of next generation sequencing. So you may be familiar with this face, Rob Mitra, who was on my thesis committee, actually. When I was a graduate student, he just started as a junior faculty back then after his graduate work with George Church. So Rob really did the pioneering work. Essentially, he did this they call polymerase sequencing. They really have a single molecule in a polyacrylamide gel so that you can amplify that signal so that you can do sequencing by synthesis. So you can really, it's a prototype of next-gen sequencing. And then, of course, at the time I was finishing up my PhD in 2005 and work done by Jay Shanduri, Greg Parika, two graduate students. Of course, Rob initiated a lot of that in the beginning. So that really is kind of the larger scale of polymerase sequencing of E. coli genome. So that really suggested the revolution of next-gen sequencing is going to happen. And so I really seized this opportunity and moved to George Church's lab. So I've done a lot of things in the lab, essentially to develop genomic technologies. Some of that became kind of the prototype of exome sequencing you probably heard of. So one of the things I used this kind of targeted sequencing approach was to identify RNA editing sites. So the reason I work on that is because I heard about that when I was a graduate student. And also, people like Jason Chenjura and Kun Zhang, two more senior postdocs, they already said, Jason, I'm going to work on exome sequencing. So there's wasn't much left for me to use that technology to apply to some interesting biology. So RNA editing was just something I heard about. Maybe I can just work on that. And then for looking into that, at that point, people only knew about maybe less than 20 sites in humans and mice. And then, by using this kind of a targeted sequencing approach, we computationally predicted these editing sites first using these human EST sequences. And then we identified a few hundred these new editing sites. So that was really the reason I worked on that. I didn't, even back then, when the paper was published, I wasn't really thinking seriously about working on RNA editing in my future lab, although I was hoping to start my own lab at that point in 2009. So the one reason I decided to focus on RNA editing in my own lab was really based on genetics. So work done by pioneers in the field, Peter Seberg and Kazuki Nishikura, for example, they've really made these knockouts of the key RNA editing enzymes, ADAR2 and ADAR1. So if you knock ADAR2, the mouse will die with seizure phenotypes within 3 weeks. And then also, Peter Seberg's lab found the key substrate of ADAR2 is this mTOR receptor called gluten receptor 2. So there's a single site called QR site, essentially a single editing site will lead to a single amino acid change from Q to R. So that single change is critical for life. So that is the reason without editing of that single site, the ADAR1 knockout, ADAR2 knockout mice will die. And then ADAR1 seems to be even more important. The mouse will die before birth. But of course, we had no idea of, what's the key substrate of ADAR1. And that's probably why the paper I published in Science as a postdoc was kind of important, because that would generate a lot of candidates. Maybe some of that will be key sites for ADAR1 editing. But it turned out we were very wrong in terms of finding the key substrates of ADAR1. So I'll tell you that in a second. All right, so then when I moved to my own lab, I started my own lab. So unlike many other fellow trainees, postdocs, you already have maybe many years of foundation of working on maybe RNA editing as a topic. I really didn't do much on RNA editing, except that one paper, just using some genomic tools to identify editing sites. But eventually, I think the genetics paid off. So in this work we did together with Carl Walkley, my close friend and colleague in Australia. So what we did was two things in this paper. The first thing we did was to show this ADAR1 knockout mice, the phenotype, the mouse died before birth, is really RNA editing dependent. So in this work, we first generated a knock-in mouse by making this E2A mutation that had been known to be the key catalytic residue of ADAR1. So if by doing that, you don't have any editing by ADAR1 whatsoever. So then the mouse will die, really phenocopies the ADAR1 knockout mouse. So that really says editing is very important. But to our surprise, in this mice, well, we observed a huge amount of interferon response. So then, of course, we were scratching our head, wondering what's happening. And then eventually, we realized that without editing by ADAR1, there will be a lot of cellular long double-strand RNAs, which, by the way, will be the main substrate of ADAR1. Although my paper as a postdoc was more looking for the protein coding, like recoding sites. But vast majority of that, we're talking about maybe millions of them, are in the long repeats. Essentially, you have two repeats that are inverted in the same transcript that would fall back, right, to form very long double-strand RNAs. So without being edited by ADAR1, these long double-strand RNAs will resemble the feature of viral double-strand RNAs to trigger this double-strand RNA sensor, which is a RIG-I homolog called MDA5. So then if you knock out MDA5 in this ADAR1 editing deficient mouse, the mouse can live a normal lifespan. That really tells you this we call ADAR1 double-strand RNA MDA5 axis. In a wild-type condition, our own cellular double-strand RNAs have to be properly edited. Otherwise, without being edited, these long cellular double-strand RNAs will really activate MDA5 to trigger immune response. So now you can imagine the key substrates of ADAR1 probably will not be the protein coding events, because for MDA5 rescue, it's all about long double-strand RNAs. So the field is now really facing a challenging problem that is to find which long double strands are really the key substrates of ADAR1 to avoid being activated by MDA5 after ADAR1 editing. So I'm not going to get too much into that. We have some ongoing work and some published work. So in human genetics, it really kind of echoes what we found in mouse. So people have found, others actually have found ADAR1 loss of function mutations or MDA5 gain of function mutations in some very rare autoimmune disease. There's something called Aicardi-Goutieres syndrome or AGS. So either way, there will be more elevated double-stranded sensing. So then a couple of years ago, we published this work really spearheaded by a former postdoc, Chen Li, who's just actually started his own lab at Penn Genetics. So in Chen's work, essentially what we found is that for common autoimmune or inflammatory disease, we probably cannot afford heavy mutations in ADAR1 or MDA5. That will be too penetrant. But instead, patients could have maybe too many of the genetic risk variants as defined by genome-wide association studies, GWAS. So these risk variants in the same patient, each one of which will impair the editing of the cis-linked double-strand RNAs. And then these insufficiently edited double-strand RNAs will potentially activate MDA5 because we know the mechanism pretty well. So that could be the initial trigger of the inflammation. So without getting into a lot of the data, let me just show you something probably easier to visualize and understand. Essentially, what we've done is just look for the genetic variants that are associated with editing level differences of many of the editing of the long double-strand RNAs. So to our surprise, what we found is that these genetic variants affecting long double-strand RNAs are statistically enriched in GWAS signals for many of the common autoimmune and inflammatory diseases. Shown here is just inflammatory bowel disease, IBD, which is a common autoinflammatory disease. So if you look at the GWAS of this IBD, there are about 250 loci being mapped. We found 78 of them overlap with the long double-strand RNAs. Furthermore, we showed that the GWAS define the risk variants. They tend to be associated with the lower level of editing of the double-strand RNAs. So that makes a lot of sense because we know only the lack of editing of these long double-strand RNAs will potentially be dangerous to trigger MDA5 activation. So in a sense, we actually have seen a directional effect of so many of the GWAS loci that are co-localized with double-strand RNAs. So that directional effect is fully in agreement with the mechanism. And then because of that directional effect, we believe this can be an angle of kind of actionable human genetics for complex disease, because all these genetic risk variants will activate the MDA5 activation. And you can imagine by dampening this double-strand sensing pathway, such as MDA5, that can be a great approach. I'm sorry, I don't know what happened at the end. Oh. Yeah, I'm sorry for the internet problem here. Okay, so yeah, so we really see this directional effect. So now, in some more recent, unpublished work, what we are hearing, I don't have a lot of time to go into the detail of the data. What we have done here is to take, the single cell RNA-seq data from the islets of, type 1 diabetes patients and healthy individuals, and also we have some, so-called pre-type 1 diabetes patients. Essentially, these individuals do not have the disease yet, but they have autoantibodies present, suggesting they have a high chance of developing type 1 diabetes later on. We also have the whole genome sequencing data of these individuals. So what I'm trying to show here is that the x-axis is essentially the polygenic risk score by combining all the genetic risk variants at the GWAS loci that overlaps with the long double-strand RNAs. So if you have that polygenic risk score that I call double-stranded RNA burden, on the y-axis, we also have the single cell data to measure the interferon response in different cell types, but in particular, we see much higher interferon response in beta cells in patients. So if you do that, we actually can see a very nice positive correlation between the genetics predicted polygenic risk score or double-stranded burden with the interferon score. So we believe this interferon response should be MDA5 dependent, which is ongoing work in our labs. And then in the future, what we envision is that we can use genetics to stratify the patients with higher genetic burden of double-strand RNAs. And then for these patients, perhaps we can stratify them and then treat them by antagonizing the double-strand RNA sensing pathway. All right, so very quickly, and I want to switch gear a little bit. Great, so now I try to focus on the biological functions and mechanisms of RNA editing. Essentially, that is to avoid MDA5 activation. That has a lot to do with autoimmune disease. I didn't have time to talk about cancer. ADAR1 actually becomes a very interesting immuno-oncology target. I'm involved with a company on that as well. But the other aspect of my lab, which also kind of turns out to be very interesting and a kind of lucky situation, if you think about ADAR editing substrates, although I said most of the ones are very long double-strand RNAs, but there's still hundreds of these double-strand RNAs that are edited in a very specific fashion in a short kind of hairpin double-strand RNA. So then the idea is that can we try to mimic what happens in nature like this by engineering something so that you can really edit, let's say if you're the adenosine in a linear mRNA, you want to edit, maybe that A is the G2A mutation, you want to edit that A back to G. So then we can deliver an antisense oligo, maybe RNA oligo, that will base pair the target mRNA in this kind of artificial double-strand RNA fashion. So by doing that, we showed that we can recruit the endogenous ADAR enzymes to make this very specific and efficient A2I or A2G change. So then, based on that work, together with Thorsten Scheffel's lab in Tübingen University in Germany, we actually co-founded a company maybe around 3 years ago. So there's a lot of kind of additional developments in the company based on this kind of a simple approach. So one thing we did, even before we started the company, is to demonstrate the feasibility of this approach using a lot of in vitro data, which I don't have time to show you, but also in vivo data for treating kind of a preclinical development of treating this disease called alpha-1 antitrypsin deficiency, or AATD. For this particular disease, this gene called SPINA1, almost all patients have the same mutation, that is a G2A mutation that will lead to a single amino acid change called E2K change. So as a result, this protein, this transcript is produced in the liver, and then for the resultant protein will need to be a misfolding problem. This protein will be stuck in the liver, but this protein's function is actually not important in the liver. It is important for this protein to be not misfolded, obviously, to get to the lung through the bloodstream. So you can imagine with this mutation, these proteins will be stuck in the liver. They're going to have a liver fibrosis problem, and also they have a lung disease, like a COPD type of a disease. So editing this mutation from A back to G, obviously, will be a great approach. So one thing we did, really spearheaded by two former postdocs, both of whom are now working in the company, Paul and Inga. So what we showed here is to deliver this chemically modified antisense oligo drug into this humanized AATD mutant mice. So what we showed, is that now we really can achieve, with 3 different versions of oligos, we can really show efficient editing. And also another marker is to check the concentration of this enzyme AAT in the bloodstream. So we really can show we can pass the therapeutic threshold, which is that kind of dashed line. All right, so just to summarize this part, we really believe this RNA-based editing approach can really borrow a lot of ideas in the RNA therapy, such as siRNA, such as chemistry, such as delivery. But of course, compared to CRISPR-based therapeutics, which is really revolutionary, it's one and done kind of approach. But of course, the safety has been a very big concern in the field. So we think, of course, our editing capability is much lower compared to siRNA. We can do some editing work. So then we can really think we can combine the merits of 2 different types of approaches in this RNA editing modality. So using this approach, we believe this is not going to be limited to fixing some of the G2A mutations often seen in rare disease. Perhaps what excites me most will be making A2G changes to maybe treat common disease. You can imagine there will be targets by making a single A2G change. You can make a single amino acid change that may be a post-translational mutation site. Then you can activate or deactivate that resultant protein, particularly in the context of activation that is not easy to develop, even for small molecules, for agonists. Right. So with that, I really feel the field that I am very lucky to be in on editing has really gone a long way. By understanding the biology and the innate immunity. So now we really think that pathway is so relevant for cancer and autoimmune disease. And of course, using this kind of a simple, double-strand kind of feature, we can, and the endogenous ADAR enzyme that can be a great therapeutic tool as a platform. So yeah, so with that, I just want to, have two major points I want to mention. One thing I realized that since WashU, I've been in the genetics department at Harvard and at Stanford right now. So you don't have to be in the genetics department, but I think the genetics is really the guide in my career. I didn't fully appreciate that when I was a graduate student, but now I really think that can really tell you there's something important. Genetics doesn't lie to us. So it's great, I trusted that, but as I mentioned, I also feel I've benefited a lot by being able to combine this computational and experimental dry and wet approaches. And I would also once again emphasize this computation is not just facilitating something, it can be a driver in many cases for interesting questions. Yeah, with that, I just want to thank my amazing group, and also want to thank Gary again, and congrats to you. That was 2005. Thank you very much.
Hello, great talk. Thank you so much. Do the sequences that are substrates for ADAR1, the sites, do they tend to have a common function, or are they just random sequences that can form dsRNA?
Yeah, so we actually look at, well, essentially most of the double-strand RNAs at the GWAS loci, they're located in the 3' UTRs of host genes. Now, being in the 3' UTR is important for any double-strand RNAs that can be potentially dangerous. They have to be in the cytoplasm, because that's where MDA5 is as a cytosolic double-strand sensor. So then what we actually try to look for the genes that host these long double-strand RNAs, typically in the 3' UTR, and look for Go terms. We actually didn't find any functional enrichment, although there are some interesting exceptions, interesting cases. So I think the message here is that if you look at the GWAS for the protein coding functions, you're going to totally miss out this particular mechanism.
So that's the correlation between the double-strand RNA with the inflammation. This relationship is context dependent, or it's cancer type dependent. Why you focus on the liver? Yeah.
Well, I think maybe you got a bit confused. I was talking about almost two very different things about ADAR. One is about ADAR's biological function in suppressing MDA5 dependent innate immune response. The other is to harness this endogenous ADAR as a platform for transcriptome engineering. That was done in the liver for reasons of delivery. It's just more feasible to deliver oligos, for example, into the liver. So for the innate immunity, that we find, the ADAR1 is ubiquitously expressed, particularly maybe just because having high expression of ADAR1 is quite important to make sure we take care of or label our own cellular non-double-strand RNAs, which, by the way, is very abundant in almost all cell types. So that is very important. Without that, we're going to have a lot of innate immune response.
Hi, my question is, how conserved is ADAR in evolution? Does fly have them? Does yeast have them? That also about MDF1, right? If its functions to suppress repetitive sequence, and then maybe some organisms don't really have much repetitive sequence, won't have that. Or maybe some organisms have repetitive sequence, but they don't have the MDF1, whatever.
Yeah,
and then maybe they don't need it, right?
Yeah,
they don't have immune response.
It's a good question. Obviously, I didn't have time to get into a lot of background and data. So ADAR is conserved in metazoans. Pretty much all animals have ADAR, but even for animals, there are different scenarios. In mammals, there are two enzymatically active ADAR enzymes, ADAR1 and ADAR2 I mentioned. These are very conserved in mouse and humans, and also, of course, in primates. But in Drosophila, for example, there's only one ADAR enzyme that is the closest homolog to ADAR2. There's actually no ADAR1. And also, it's actually quite interesting. In Drosophila, you actually barely have very long double-strand RNAs that are formed in mature mRNA. So, if you look for any long kind of inverted sequences, either repeats or anything, you barely see any of the long double-strand RNAs that can be formed. The control is that if you look for tandem sequences in the mRNA in Drosophila, you actually see a lot of them. So that's actually a huge contrast. We still don't fully understand why, but maybe just because you don't have, because basically somehow you don't have ADAR1 in Drosophila. As a result, you cannot tolerate many of the long double-strand RNAs to be present, because that will be a big, big problem. So during evolution, the reason we can, well, in humans and mice, we also know there's a lot more tandem repeats compared to the inverted repeats. That suggests at the genome level, there's a mechanism to purge, not entirely, these long double-strand RNA formation. Now, it's not entirely purged, maybe because of the presence of ADAR. We believe ADAR probably evolved before the presence of these long double-strand RNAs that are often formed by repeats. You probably know during evolution, these, even between human and mouse, we say they're very conserved, pretty much for the protein coding parts. For the repetitive elements, human mice are not that similar. There's a lot of kind of a bombardment of these transposable elements independently in these two different species. But the reason you can tolerate many of the long double-strand RNAs maybe is because of the presence of ADAR1 in the first place. Then, of course, what's the ancestral function of ADAR1? That's another interesting question. Many in the field are trying to pursue.
Okay, let's thank Billy again.